Welcome to this CME certified program entitled Narrowing the Gap, Understanding Biosimilars. I'm Dr. Leonard Calabrese, a professor of medicine at the Cleveland Clinic Lerner College of Medicine here in Cleveland, Ohio. And joining me in a little bit will be Dr. Edward Li, Associate Professor, Department of Pharmacy Practice at the University of New England College of Pharmacy in Portland, Maine. These are our disclosures. And these are our learning objectives. They really involve recognizing the process of building biologic, recognizing the manufacturing difference between biosimilars and the originator, describing the FDA pathway to the approval, which we call the totality of evidence, and then considering what impact these will have on our practice. So I'm going to lead the way here in talking about the difference between biosimilars and reference products. I suppose that the first question that we ponder when we discuss biosimilars is, why do we have them? If they are truly similar to the originators, what are the advantages that may accrue to our patients or our society? There's no doubt that biologics have raised the bar in the treatment of many diseases, of which a large part of them are immunologic diseases. They have offered new standards of efficacy to many of our patients. At the same time, these are extraordinarily expensive drugs. And the costs of these drugs have really only increased dramatically since their approval. The introduction of biosimilars, particularly in Europe, has led to dramatic reductions in some countries that have socialized healthcare systems with one payer, as much as 60%. There are estimates that biosimilars may lead to savings of over $60 billion as we move ahead. The first question that we will kind of sneak into in terms of a process is to answer the question of what is a biopharmaceutical. Biopharmaceuticals are really any product from a living organism that are applied to healthcare settings that may modify some aspect of a disease in terms of prevention, treatment, etc. I think that a more apt question might be, what is a biologic drug, which is a subset of biopharmaceuticals. And these are genetically engineered proteins derived from human genes, often expressed in eukaryotic or prokaryotic cell lines. The ultimate product are therapeutic proteins, which are then put into practice. Biologics are large protein-based therapeutics that vary in size and complexity. I like this figure because it contrasts the small molecules over here that you can see an aspirin moiety compared to the large polyprotein immunoglobulin at the right side of the slide. Complexity is dramatically increased over the span of evolution of these types of compounds. And I think it speaks legions for why biosimilars are not carbon copies of the originators, such as generics are to these small molecules. Another question is why it's important to understand the biosimilar development process, because really this is at the core of what a biosimilar is. As we will point out in the next few minutes, the DNA sequence is of public record of these proteins that we are used as biologic therapeutics. However, aside from that, much of the rest of the process is proprietary, and we'll get into that in a more granular fashion. So at the end of the day, the biologic, the originator compound, the ones that have been approved by traditional pathways, are unique macromolecules. So we have to go into this development process a bit more. Biosimilars are a sort of copy of commercially available biopharmaceutical, which we often call either the originator or the reference product. These are developed after these drugs come off a patent. At 30,000 feet, I will tell you that these biosimilars, which are approved, have undergone rigorous analytical and clinical assessment and have met all the requirements of the regulatory agencies for the biosimilar pathway. We like to use the term highly similar because that is in the pathway, and I'll give you a more granular view of what that is in the next few slides. In terms of cautionary notes, biosimilars are really not what some people refer to as biobetters. A biobetter would be an originator drug that has undergone modification to enhance it, that actually are structurally different from the original licensed biopharmaceutical. And in that token, it is really a different drug. From the first chimeric TNF infliximab, developing human antibodies such as adalimumab would be part of this evolution. Generic drugs, as I told you, are literally carbon copies of small molecules, and they are regulated under different legislation. This is a complex slide that I really don't want to go into any depth, but what it points out is that there are multiple pathways to biologic drug approval: small molecules, generics, biologics, and biosimilars. Each of these have their own regulatory components and their own detailed pathways for approval. We'll enumerate them as we move along in the following slides. The goal of standalone and biosimilar development are actually quite different. The originator compounds are developed based upon developing a biologic therapeutic by creating these express proteins as I have previously described. There is an analytic program which characterizes them. There's, of course, toxicology, there's pharmacokinetics and pharmacodynamics, but the bulk of these programs are robust clinical studies that must go through first in man, phase 1, phase 2, and then phase 3, which are rather expansive. On the other hand, the biosimilar pathway is somewhat abbreviated in terms of its clinical pathway, but as you can see and is graphically depicted, the analytics of this are far more robust. They have to demonstrate this fingerprint similarity to the biosimilar in terms of its physiochemical, biologic, and immunologic properties, and then go into non-clinical studies which are highly abbreviated, and then the clinical programs are quite small. And as I'll show you, and as we'll talk later, with the approval pathway of extrapolation, only limited numbers of diseases have to be looked at for full therapeutic across the approved spectrum of the reference product. This is a nice flow diagram that kind of depicts how biosimilars evolved. We start with the DNA sequence of the reference product because that is a part of public domain. This DNA then is inserted into a vector that will allow it to be used to transfect host cells to grow the protein. At this point in time, the vector is proprietary, the host cell line is proprietary, and as we move down to look at the fermentation and the actual process of cultivation of these cells that will be the actual machines for biosimilar, all of this is proprietary in terms of time, pH, temperature, media conditions, all which go into influencing post-translational modification. There are numerous processes in the expression of the protein, purification of the protein, and the stabilization and packaging of the protein, which can influence the biologic properties of the biologic agent. So the term reverse engineering is often applied. Reverse engineering means that we know what the DNA is, but we must develop a product that matches up to the reference product in virtually every way in terms of its physiochemical properties, pharmacokinetics, pharmacodynamics, immunogenic properties, and these are evaluated at each and every step of the biosimilar evaluation process. Each step has critical ramifications in the drug development. We also use the term stepwise approach. If you look at preclinical phase 1 and phase 3, the largest part of this program is really preclinical. These in vitro studies really identify the physiochemical similarities between the biosimilar and the reference product. They determine if in vivo studies are needed. It's really not mandated that in vivo studies and preclinical models be performed if the entire package and totality of evidence is looking good. The phase 1 studies are really the most important, where these drugs have to match up, particularly in PK, PD, and immunogenicity. And then finally, phase 3 studies, they will pick a disease that is an approved indication and demonstrate non-inferiority and a similar safety signal. The general principles of biosimilarity, I think I've already gone over, but it revolves around the ex vivo studies and then the clinical efficacy and safety. These are not intended to be superior or inferior. They are intended to be equivalent, and that is what drives the design of these clinical studies. No differences in safety or efficacy are expected or tolerated in this biosimilar approval process. Just to give you a snapshot of the extensive analytical characterization that is used, there are tools which can outline the primary structure. There are higher order structures that can be identified by sophisticated physiochemical techniques. Certainly, there are innumerable tests that are applied to demonstrate biologic function, and this has to do with immunologic function. These are particularly for those things that are monoclonal antibodies. Do they have appropriate FC binding? Do they have appropriate avidity? Is there some symmetry to the glycosylation pattern? And will this affect their PK or PD? Obviously, impurities from host cells have to be eliminated. And then finally, the finished product has to have a similar pattern of excipients that have been added to the reference product, so it's not altered in any type of vital way. So here we see biologic function used to establish a high degree of similarity, and I've already mentioned these: target binding, avidity, complement-dependent cytotoxicity. Will these induce target cell apoptosis? Is there FcRn binding, which would influence its metabolism and in vivo half-life, Fc binding, and ADCC and beyond. So a tall tale and a tall task ahead. Finally, no discussion of biosimilars would be complete without at least mentioning immunogenicity. This is the ability of a particular substance, such as the biosimilar and some component thereof, to elicit an immune response in the host. We call these anti-drug antibodies. These have been correlated with both toxicity and efficacy. This little cartoon points out that on the left, we have a purely xenogenic antibody, which could be of mouse origin, rat origin, or something, highly immunogenic. The first biologics were chimeric. That means that largely in a human, but the FAB and variable regions may be of non-human origin. Certain compounds are humanized. That means that they're predominantly human, but still have usually just the hypervariable regions expressed. And finally, we now have totally human, which means they meet a certain regulatory standard that there is a minimum or paucity of xenogenic proteins. Here is some data of a biosimilar infliximab looking at CTP13, looking at immunogenicity. And here you see the originator or the reference product and the biosimilar, and these lines are virtually identical. The totality of evidence is often referred to as fingerprint similarity. And here you can see mass spec analysis, all these higher order analysis of complex biologic data, bioactivity, patterns of glycosylation, which are not proprietary, and then patterns of impurities, all which are looked at. This goes to make up what we now call the totality of evidence, ranging from structure, function, non-clinical studies, all the PK/PD, and then finally clinical effectiveness and immunogenicity, and that will determine whether there's any similar product of risk reduction or pharmacovigilance that will be needed. So this is the groundwork of biosimilar drug development. I'm now going to turn this over to my colleague, Dr. Edward Li, Associate Professor at the University of New England College of Pharmacy, to address how biosimilars will impact clinical practice.